início a moderação e discussão dos resumos
We're going to
selecionados.
begin the discussion and moderation of the selected summaries, stage 2. I will introduce the presenters. They're all women that are going to present in this session, and I'm going to talk about the title of the work. Xiomara Alexandra Gaetan from the University of São Paulo. Stefanie Batina Astro, also from the Federal University of São Paulo. Jessica Rafaela dos Santos Alves from the State University of Campinas. And Thais Baruel Vieira from the Federal University of São Paulo. Please, girls. My name is Aniele Pina from the Osvaldo Cruz Foundation. This is Gisele Mello, who is here with me. And each one of you has 6 minutes for the presentation, and then we're going to open for questions. We'll start with Xiomara Alexandra Gaetan, whose title is The Trump CCS is Essential for the Transmission of Zooids of Plasmodium burgei. You have 6 minutes, and we're going to warn you when you have 2 minutes.
Okay, so hello everyone. As all you know, Plasmodium zooids are a motile form of the parasite, highly specialized in structure, shape, and molecular machinery to invade host cells. The mechanism that ookinids and sporozoites use to invade cells are poorly understood, but the merozoite invasion has been well characterized. So during the Plasmodium erythrocyte cycle, specifically during the reorientation phase, exists the participation of the PCR-CR complex that I'm going to call in this talk as TCR-CR. This complex was initially described in Plasmodium falciparum merozoite, involved the participation of the protein TRAMP, CSS, RIPER, SIRPA, and RH5. All these members of the complex are essential for the merozoite blood cycle. Orthologs of this protein are present in Plasmodium knowlesi and Plasmodium berghei, and is importantly that in a sporozoite has been found transcript of TRAMP and CSS. So with all this information, we propose to evaluate the role of TRAMP and CSS in the sporozoite using the murine model Plasmodium berghei. So initially, we construct a Plasmodium berghei CSS hemagglutinin parasite line using this strategy. And with the sporozoite of this parasite, we perform co-immunoprecipitation assay followed by mass spectrometry analysis. So our results reveal that CSS appear as a protein indeed in a sporozoite, interacting with TRAMP, with protein TRAMP, and it's important to mention that RIPER was not found in the CSS interactome. So with this result, we confirm that these two proteins are present in the sporozoite as a protein, and they both are interacting together, forming a complex. So the next step was to evaluate the role of these two proteins in the merozoite. So to achieve this objective, we construct a conditional knockout of CSS and TRAMP, is TRAMP here, using the dimerizable recombinase system, using these two strategies here. The importance of this system is that we induce this thing is not working. The important thing that we can induce gene excision when we put the rapamycin in the parasite. We also use internal control of each parasite that were untreated. So with this, once we obtain this both parasite, we infect mice and we fed mosquitoes Anopheles stephensi on them. And one day after the feeding, we start the rapamycin treatment in the sugar water until day 20, when we collect the salivary gland sporozoite to perform all the experiment. So we didn't observe difference when we analyzed the amount of sporozoites from salivary glands in both parasite lines, regardless the rapamycin treatment. The next step was to test if this sporozoite have the ability to infect hepatocyte. So the cell traversal assay show us no difference between both parasite lines when we compare with the control. It means that this important migratory process is not affected by the knockout. But however, when we analyze in the knockout population in both parasite line, we observe a reduction in the ability to invade cells and a very strong impairment in the development of this parasite inside the hepatocyte. Microscopic analysis of this time point show us that in the untreated parasite, we observe a very clear formation of the Parasitophilus vacuole when we stain with US4, clear in blue here in the CSS parasite, and in magenta in the trap knockout, in the trap population. And when we analyze the knockout population, we observe a very diffuse formation of the Parasitophilus vacuole, observed but a very soft staining of the UES4 marcation. We also observe parasite in the nucleus, that this is this staining grade, and also presenting the same phenotype with a very malformation in the Parasitophilus vacuole. So with all the results, we can conclude that the knockout of TRAMP and CSS leads to a similar defect in the sporozoite stage and several impairment in the host cell infection. We also can conclude that the TC complex is dispensable for the sporozoite development inside the mosquito and for the cell traversal process of the sporozoite. Thank you.
Vou chamar agora a Stefanie Plastina.
Now I will invite Stefanie Plastina Astro from the Federal University of São Paulo. You have 6 minutes for your presentation.
Apresentação da Stefanie, por favor.
Stefanie's presentation, please.
Boa tarde. Hoje eu vou apresentar dos
Good
braços
afternoon. Today I'm going to present one of the branches of my doctorate project on the mapping of gametocyte development of in vitro cultured Plasmodium knowlesi by single-cell RNA sequencing. As we all know, the knowlesi Plasmodium belongs to the same branch as the Vivax, and it's very similar to Vivax in comparison to falciparum. That is why it becomes an interesting model of study of Plasmodium vivax, and also it has been demonstrated as a potential parasite in the Asian region for human infection. And speaking of the development of gametocytes, which is my work, we know that the Plasmodium falciparum has a development of gametocytes which is a little bit slower, from 10 to 12 days, while Vivax and Nosci need a few hours, between 36 and 72 hours. The development mechanism of these gametocytes is still not well characterized, so the goal of my work is to characterize this development through single cell. In order to do this, I cultivated the parasites in a culture. I did the per-cult to reduce the number of non-infected cells, and I captured these parasites in the hive system, preparing libraries that were sent to sequencing and that generated the results of my the data of my analysis through our studio, Python doublets, and PlasmoDB. The first result that I obtained was this mapping with all of you map with the stages of the parasites, and especially the clusters that I was interested in, which are the probable gametocytes 1 and 2. So I selected these clusters and I did a reclusterization, because with the reclusterization I am able to understand in a better way the pathway of these cells and infer their development. So my reclusterization gave me 7 clusters with a total of 1,456 cells, and giving you a spoiler, 48 mature female gametocytes and 21 mature male gametocytes. So I started the analyses. Here I included only a selection of genes because there were many, but I analyzed the genes that were up to 10 times more expressed, at least up to 10 times more expressed in each cluster. And I compared and analyzed with the information that I had and we have had about falciparum and Vivax, and the information available at PlasmaDB, so I could infer something about their development stage. So I was able to mark the stages. I reached 4 stages before full maturing, which were stages 1, 2, 3, and 4. And the difference between them is the quantity of genes in the erythrocytic cycle. So stage 1 has many more genes being expressed of the erythrocytic cycle than 2 and 3. And stage 4 basically does not have genes of the erythrocytic cycle. It has genes of gametogenesis, but it has more male and female markers, so we cannot infer yet if that is a female or male gametocyte. So I mark them as the last stage before complete maturing.
Here, I've selected 4 markers. I chose them as good gametocyte markers, not necessarily female and male. I'm talking in general. 2 of them are unknown function proteins. One of them is PK25, which is well known, and PF25 and PV25, and also zinc finger, which is a protein. And we are able to see that these genes are not well expressed during the erythrocytic cycle, and they are more present in terms of gametocytes. Just to conclude, I was able to map the gametocyte development. I was able to identify the intermediate stages. I was able to propose the potential molecular markers, and these findings help us better understand the sexual differentiation of the gametocytes and maybe find a good marker for future treatment to block malaria so that we can eradicate malaria from the world. Tomorrow I'm going to present my poster, and if you want to ask questions or talk more, I'm going to be there. This is my poster number, and thank you for the opportunity.
Parabéns, Stefanie. Então, a gente
Congratulations, Stephanie. Now we are going to have Jessica Alpes, and she's going to talk about comparative investigation of Plasmodium vivax in bone marrow and peripheral blood samples from individuals naturally exposed to malaria vivax in Malarian Amazon, in Brazilian Amazon.
Good afternoon. First of all, it's a pleasure to share with you some data of the project I am integrated, which aims to conduct a comparative investigation of Plasmodium vivax in human bone marrow and peripheral blood samples. So this project is guided by the hypothesis that paired bone marrow and peripheral blood samples harbor distinct parasite states that reflects in a niche-associated functional programs. So to test this hypothesis, our group conducted a challenging study cohort in Manaus involving the collection of paired bone marrow and peripheral blood samples from Plasmodium vivax infected individuals. And this paired sample strategy was integrated in a workflow that combined transcriptomic methods, including bulk and single-cell RNA-seq, with phenotypic and functional assays to investigate compartment-specific parasite states. In our data, the bulk RNA-seq revealed thus an enrichment of parasites transcriptional programs in the bone marrow relative to peripheral blood, consistent with an active development of parasites, including phosphatidylcholine biosynthesis, invasion, gene regulation, and DNA replication programs, beyond transcripts related to gametocyte development and the sexual commitment, represented here by the AP2G, the master regulator of sexual commitment in Plasmodium parasites, indicating that the bone marrow represents an enriched niche for sexually committed parasites and gametocytes. And these results motivated us to use the single-cell RNA-seq strategy to achieve a higher resolution characterization of Plasmodium vivax in this niche. And using paired samples from 2 patients, we generated the first comparative single-cell atlas of Plasmodium vivax derived from paired bone marrow and peripheral blood samples with a high comparable landscape to the reference dataset published recently. And this dataset allowed us to first explore if the AP2G expression is restricted to a defined parasite population. So our data revealed that AP2G transcripts were detected across several parasite subsets with compatible expression in both compartments, bone marrow and peripheral blood. Moreover, along the asexual trajectory of the parasite, our data revealed that the AP2G transcripts expression was gradual, as demonstrated in these pseudo-time graphs, proeminent in trophozoites and schizons, while reveal a significant decreasing in late schizons. Moreover, while along the female trajectory to late female gametocytes, the AP2G transcript expression arose earlier to late female markers including PVS25 and LAP5, represented here in this pseudo-time graph by blue and brown curves, respectively. Finally, we looked at how immature sexual markers were distributed across the tissues. And our data revealed that despite compatible expression in PVS16 positive cells across the compartments, the peripheral blood had a greater representation of PVS25 positive cells, consistent with a greater representation of late female gametocyte-associated states. And this data corroborate our functional assay of membrane-feeding assay, where mosquitoes fed with peripheral blood samples had an increased number of oocysts counts related to mosquitoes fed with paired bone marrow samples. So taken together, this data strengthened our hypothesis that bone marrow and peripheral blood harbor distinct parasite states. Our single-cell data revealed that the AP2G transcripts were detected across multiple parasite subsets, and its expression also arose before late female markers such as PVS25 and LAP5, supporting its role as an early regulator of sexual commitment in Plasmodium vivax. Moreover, we also revealed that peripheral blood showed a greater representation of mature female gametocyte states represented by PVS25 transcripts, and this transcriptomic data was supported by functional assays of membrane-feeding assay, where mosquitoes fed with bone marrow samples showed a lower infection outcome related to peripheral blood. So thank you all for attention.
Obrigada, Jéssica.
Thank you, Jessica. Now we are going to call Thais Vieira from the Federal University of São Paulo. Her title is Transcriptional Differences Between Salivary Gland and Saliva Isolated Plasmodium vivax Parasites Revealed by Single-Cell RNA Sequencing.
Today I'll be presenting the work we've been doing with Plasmodium vivax porozoites and the difference between their transcriptomes from the ones collected from the salivary gland and the saliva. So our data was collected from mosquitoes 14 days post-infection. Our mosquitoes were infected by human samples, and we prepare a library using the Hive technology. For those unfamiliar with it, it uses gravity for single-cell isolation as opposed to 10x, which uses microfluids, and it's a very interesting system. So we prepared the library, we sequenced the single cell, and we analyzed the data. The first thing we had to do was to check if the data coming from the different origins would cluster separately, so form two different clusters. Here we see that after we integrated the data, we got 4 different clusters. So all of the analysis done that I'm going to be presenting will be from clusters 1 and 2, so the pink and the yellow ones. Clusters 3 and 4 are for a later date. We're still working on that. So here, I just want to prove to you that what we got is not a batch effect, and it's really biology. So here we can see it's the same UMAP from the previous slides, just separated from origin. So here are the cells that were collected from the salivary gland, and those are the cells collected from the saliva. And we can see that they are mixing in the clusters. So there are some cells with salivary gland origin clustering with the cells from saliva and vice versa, but there are far more cells from the saliva clustering with the gland, and that suggests to us that there is a directionality in the maturation of the sporozoites. So to prove that, we performed pseudotime, which basically gets the genetic profile from each cell and fits a curve onto it to show us the probable development, where the cell is probably going. So here we can see, again, the same graph, but the colors, the purple are the more immature cells, and the yellow are the more mature cells. So we can see that there is a kind of path going from the salivary gland to the saliva. And again, in case you still do not believe me, we perform an RNA velocity analysis, which uses mRNA degradation to check the velocity of a gene or how much that gene is being expressed at a given time. And that will give us where is this gene, that cell is probably going. So this cell is probably becoming this other cell. So we can also fit a path onto those cells. So here on top we have the salivary gland, and on the bottom the saliva. The arrows pointing here in the salivary gland, they're not big, which tells us that there's not a lot going on here in the salivary gland. But once they get closer to the saliva, we have these beautiful arrows pointing towards it, which again gives us a path going from the salivary gland to the saliva. So once we have the pseudotime, we organize the cells in pseudotime, and we looked at the genes that are being expressed, and we found that the saliva cells are expressing far more invasion and motility genes than the salivary gland. So here from left to right, we have the pseudotime, with right being the most mature cells, and black is the peak expression of each of those genes. So we see, for example, here GAS8, which is a motility gene that has its peak on the cluster that represents saliva. So in conclusion, we can see that there is a non-negligible difference between the cells collected from the salivary gland and the saliva, and that can be instrumental for us looking into targets for potential vaccines that are targeted for Plasmodium vivax, because we usually use sporozoites from the salivary gland to do our studies for vaccines. And if anyone wants to talk more about it, I just presented my poster, but I'll be around. And on Monday, we have a poster session again. This is the number of my poster. And thank you for your attention. Thank you for the opportunity.
Parabéns, meninas, pelas apresentações.
Congratulations on your presentations on time, and we're going to have space for a single question.
Bom, então a gente convida todo mundo para ir ver os posters
Now
das
we invite you to see the posters that are in this National Malaria Research Meeting. Thank you all.
Boa noite, todo mundo.
Good evening, everyone. This is one of the most interesting tables, last but not least. You're all a little bit tired, but I wanted you to give your full attention to this table. It's a table on drug discovery, and we have renowned speakers, and let's start now.
Boa tarde, everyone. I'm glad you made it to the last table. I would like to invite Brice Campo. He's the head of biology and modeling, and he provides scientific advice and leadership to a portfolio of drug discovery projects and pharmacology platforms. He works with partners from both academia and industry to deliver pertinent and optimal biological test cascades and modeling simulations, as well as a tolerated and efficacious preclinical candidates that will form part of the next generation of antimalarials. We would also like to invite Purnima Bhanot. Yep, please. I would like to invite now Purnima Bhanot. Purnima is a faculty in the Department of Microbiology, Biochemistry, and Molecular Genetics at the New Jersey Medical School. Her research program focuses on malaria parasites. Malaria causes upwards of 500,000 deaths a year, mostly in young children in sub-Saharan Africa. The Bhanot lab studies the mechanisms by which the parasite infects the liver, from where it launches a disease-causing infection of red blood cells. Her goal is to develop drugs and block the parasite's development in the liver and prevent malaria.
Well, I will keep inviting the people over here. The next one is Rafael Guido. I have got short CV here, otherwise it will take longer. Rafael Guido is an associate professor of the Institute of Physics of São Carlos, University of São Paulo. His expertise lies in integrating computational and experimental methods, focusing on structural elucidation of molecular targets to support delivery and development of new drugs. Please, Rafael. Finally, I'm inviting Célia Garcia. She's a full professor of the University of São Paulo in the Department of Clinical Toxicology Analysis at the School of Pharmaceutical Science. Her focus on how chemical science modulate pathogen-host interactions. Her main research lines include screening natural and synthetic compounds for malaria, for antimalarial drug development. Please.
Okay. Hi, everyone. First of all, I'd like to thank the organizer, Carol, Daniel, for inviting me. It's a pleasure to be here. It's been a long time since we collaborated, never been to Sao Paulo, so it's been a pleasure to be here. So thanks for the invitation. So I'm just going to show you a little bit of what MMV is doing right now, what is our strategy, where we're going, what we're trying to do to combat some of the key issues that we're facing, such as resistance. But before I do that, I just wanted to introduce you to MMV, who we are, what we do. So basically, MMV was born in 1999 on the basis of pretty much an empty pipeline for drug for malaria, as well as a very high burden of malaria at the time. And we are now a Swiss-based foundation, as well as a US charity one. We're about 100, 110 people, mainly based in Geneva. And we work following a model of, we call a product partnership, product development partnership, or PDP if you prefer. So basically we receive funding from many areas, so governments, charities, and so on. And then we reinvest those funding into a malaria research program. So we go from early discovery, clinical phases, as well as access. We want to make sure that the drug that we develop are reaching the patient and in the field. So basically, we work with many partners. Of course, we don't have any labs, so we need partnership. That's the core of our institute. And we work with both academic research, CROs, as well as pharma partners. So as I used to call this slide, this is the rest of the team, basically. We're only 100 people. We're going to do everything. We really need the rest of the team, which are mainly spread all across the world, with about 40% of our partner in endemic area. And of course, South America and Brazil is one of our main collaborators. So with that, we've been growing our pipeline the last 20-something years, 27 years. So we have now a lot of drugs which have reached the market. As you can see on the far right, a lot of them are heavily dependent on artemisinin, and I'm going to get back to that. It's an important point. There is another one that you all know very well, tafenoquine. I'll discuss it a little bit more. Brazil is embracing tafenoquine very heavily and steering the way towards using tafenoquine probably worldwide. But we also have a lot of drugs that are under development, both in the clinical phases as well as in early discovery, and I'll give you a flavor of a bit of those ones. So despite the heavy pipeline that we have and the many drugs available, the progress in malaria have been stalling the last few years. We've been discussing that today. There's been a lot of discussion around malaria resistance, which is one of the reasons probably. But there are many reasons to progress stalling in malaria. One of them also is, of course, a reduction in the funding, and it's getting harder and harder to get funding for global health in general, as well as malaria. So we would hope that we would be on the bottom curve, reaching toward the target, a world free of malaria. However, we're more on the orangish curve. So the strategy we're putting together now, together with our partner, is how can we bend the curve and go back on a reduction trajectory. So in 2024, we published our new 6-year plan strategy. I'm a scientist, I'm not a businessman or a marketing guy, so I'm not going to bore you with that. So I just wanted to show it to you. We have a strategy that we developed together with many people. And clearly our strategy is continuing developing medicines for treatment, but as well as shifting more and more towards chemoprevention. And then using all those tools to reach elimination. We also have a lot of work ongoing right now with transmission blocking. The fourth pillar of our strategy is going beyond malaria. We've kind of slowed down on this fourth pillar right now. We just want to concentrate on malaria. Malaria is key. We're still seeing that the curve is not bending. So first let's take care of malaria, and then we will switch to other diseases if we can. So putting together this strategy, last year we had a new CEO joining the company, and we kind of discussed within the strategy again, and we kind of evaluate 5 big bets that we are really focusing right now the entire company on. 2 of those big bets are in the R&D side, and 3 of them are in the access side. The 2 first one are, oh sorry, where is it? Yeah. The 2 first one are single-dose cure as well as long-acting prevention, and I'll discuss about that. And on the access side, we want to continue tackling Vivax elimination. I'll touch on that with a slide on tafenoquine. And then we want, of course, contain resistance. And also one thing that we're developing more and more is try to establish supply chain directly in endemic countries. So we have a lot of work right now ongoing in Africa for that. So putting those bets together, putting the strategy together, of course, we needed to identify what are the key challenges that we're facing today. A few of them have been discussed today. Of course, the main one is resistance emerging to artemisinin, but as well as potentially one day lumefantrine. In Brazil, we have a lot of resistance to chloroquine, so clearly resistance is something we need to tackle. In malaria prevention, we are really limited by the choice of product available. SMC is working really well right now in Africa, but of course it's also facing resistance. And the duration of protection is not long, so it needs constant administration of treatment. There is a huge gap for pregnant and lactating women, mainly in chemoprevention. The good news is that Coartem has been now approved for a pregnant woman in the first trimester, so that's a progress. But remember, when we talk about resistance, if we lose artemisinin and then lumefantrine, then we will be back to 0 with no solution for this population. Of course, prevention of relapse is something that it talks to you a lot in Brazil, and we're doing, it's hard right now to continue developing some of those drugs because assays are not very available. However, tafenoquine is there, but it still has an issue that it cannot be administered to G6PD deficient patients. So there's still a gap there, and we are developing new drugs to try to counter that. And transmission blocking, I discussed about that. I'm not going to touch on that too much today, but we are developing low-dose primaquine to be added on top of ACTs to try to constrain transmission and also constrain resistance. So, resistance. Yes, when we talk about resistance, we always talk about parasite resistance to the drug. And of course, K13 mutation have been identified in Africa quite a few years ago. We now see also phenotypic resistance to artemisinin, at least in vitro assays. We don't see yet massive failure of treatment because the partner drugs are still active. We don't know how long that's going to last, so we are worried about that, and that leads me to the next step, which is lumefantrine resistance. We also see something cooking right now in Africa. We see some shift in IC50s. We discussed it today with several presentations. We also see something in Brazil. It's not a yet clear-cut resistance to lumefantrine, but the history told us that when the signals are appearing and start to cook, the fire is going to be there soon. So we need to run against the clock to try to find a replacement to lumefantrine rapidly. There are also other resistance. Mosquitoes are becoming resistant to insecticides. The parasite is also becoming resistant to rapid diagnostic tests. So clearly resistance is an issue, and that's something we need to tackle. One of the things that is also impairing right now treatment, in particular in Africa, and also participating in resistance, is adherence to treatment. We know that ACTs are great, but they still need 3 days of administration. A lot of patients, when they get malaria in Africa, they use the first pill, second pill, they feel much better. Sometimes they keep the third pill and they don't use it. That also participates in establishing resistance. So we are, oh, interesting. Oh, spoiler alert. We are right now working on a single-dose cure treatment, and we've made progress, and we hope to have such treatment on the market pretty soon. So what are we doing, and how is our pipeline looking like in terms of answering all those challenges? So we have, I would say, an urgent plan, a medium-term plan, and a long-term plan. The urgent plan is trying to take care of artemisinin resistance. We know it's there, it's coming, it's been there in Southeast Asia, it's coming to Africa, and it's going to hit hard. So what can we do to try to contain the resistance or continue using ACTs for as long as we can until the new drugs are coming over? So I'm not going to go through all of them in details. Of course, the slides will be available, and I'm happy to discuss about that later on, but we are, of course, using multiple first-line therapy, so basically rotating between ACTs in order to rotate also between the partner drugs. We are working on triple ACT, so adding another drug to the double combination. As I said earlier, single dose of loprimacquine is right now in the pipeline. We want to add that to ACTs to constrain the transmission of resistance. And of course, I'm sure you've all heard about that. We recently had a very nice data with Ganlum, a collaboration with Novartis that was announced last year in ASTMH. Phase 3 has been complete. It's highly efficacious, safe, and ready to go on the market. So that was the good news of last year. So we now have potentially in the next few years a replacement to an artemisinin-based combination. However, I want to note that Ganlum, there is still lumefantrine in there. So we still need to take care of lumefantrine, because if we lose lumefantrine, we will also lose this potential treatment combination. So lumefantrine, we are now in the medium plan, I would say. Those are the next generation of drugs. So we have a triple combination with Ganlum and adding a PfPKG inhibitor, cipagamine, with Novartis. We also have a new combo with Zydus in India, which is called Zydus Ferroquine. This has the potential to be the first single-dose cure treatment available on the market. And we also are working on severe malaria, because artemisinin resistance, it might not be an issue today for treatment because it's in combo with lumefantrine, but it could be an issue rapidly for severe malaria where artesunate is used alone. So we're trying to develop together with Novartis a cipagamine, which is a PfPKG inhibitor with another molecule for severe malaria. On the long-term plan, as I said, we want to do single-dose cure. And there are here listed below the potential molecules that are currently in our drug pipeline. They are all of them either in phase 1 or about to enter in phase 2, which means that they'll be available in 2030 around this time, hopefully. In discovery, we still continue to fuel the pipeline with treatment, even though we are more and more moving to chemoprevention. We want to be a 50-50% pipeline in discovery. And as you can see all along the year, the new clinical candidate going into clinical studies, they have improved their single-dose value. They went from gram scale to 100 milligram scale. And also the resistance profile has been getting better and better. So we have now a molecule that has the potential for single cure treatment, but with a low resistance generation profile. So switching gear now, second part of our strategy is chemoprevention. Sure you all heard about that. SMC is the key product right there for chemoprevention. It's been used in many million children. So here are the number for 2023. 53 million children have been dosed with SMC. 75% protection, so it's a very highly efficacious and cheap. It's very cheap to make and very cheap in terms of price. So it works pretty well on its own, and it also works very well together with the RTSS vaccine. So this is a product that we want to protect. However, resistance is also coming for this product. Resistance is developing in the south of Africa, and this product is at risk of not being active in a few years. So we need to find a replacement. On top of that, it's a short half-life product, so you need to give it several times, many pills, so you might be missing the patient because they don't always go back to get the pills at in the hospital. So, before I move to LAI, so the challenge we're facing, I talk about that, of course, the problem of the short half-life of the treatment. The vaccines are only mainly available for infants less than 36 months of age. SMC is only available for children less than 5. So all the children right now, they have no protection. So it's again another population at risk. And as I said earlier, there is no chemoprevention right now for pregnant women or lactating women. So one of the things we've been developing, and I'm sure you've heard about that because this is a topic that is very fashionable right now. So we're working a lot on trying to reinvent chemoprevention, trying to find something that is much easier to use as SMC, but still remain as efficacious and as cheap as MC. And in the last 3 to 5 years, we've been directing a lot of our effort and energy on developing a long-acting injectable. Basically, you have the profile here. The ideal drug for LAI would be to have a combination of 2 drugs that can ideally be co-injected with the same syringe. Has a long protection, more than 3 months minimum, but we are now product that probably are gearing up toward a 6 months protection. So once every season or every 2 seasons. Then you have a very high level of adherence because the patient come, get the injection, and you don't see them for 6 months. So they are protected for 6 months. So that would be ideal. No cold chain, easy to handle, and they are pretty cheap in terms of products. So really the same price than the current standard of care. We have 2 drugs in the pipeline that have now entered into a human. So it's a first. And so it's a combination of 2 drugs. One of them is a product of Atovacon that everyone knows, which is a BC1 inhibitor on the QO sites. And we have a second product, which is a new drug developed by OHSU in collaboration with us. MMV055 is an ELQ molecule. It's a new chemical entity, so it takes longer to get to human, but is now in phase 1, and we hope to complete this phase 1 by the end of the year. The goal is to combine the 2 together. We've done some testing. They don't generate resistance when they are combined together. They don't show cross-resistance. And we also see synergy between the 2 when they are added together. So in the future, this is how probably the chemoprevention portfolio will look like. We will continue to have oral standard of care, which is probably SMC, if everything goes well and we don't see much resistance. The vaccine will still be there, but there will be new generation of product coming. We are also looking at monoclonal antibodies and MMV, as well as other biologics. We work on LAI, and of course, we're trying to work on new generation oral chemoprevention. So in the last 3 minutes, just a couple of slides on tafenoquine, because as I said, tafenoquine has been very well embraced in Brazil. You guys are leading the path for tafenoquine. So it's currently being used in combination with chloroquine. It's on the label. It can only be used with chloroquine. However, we are getting worried because there's resistance to chloroquine developing. So there is a need to develop other combination with tafenoquine. And right now, some of the preclinical data we have, as well as some of the clinical studies that are being done in other parts of the world, are showing that there might be a way forward to use tafenoquine with ACTs. So of course, I forgot to say that, but it's a drug that has been developed by GSK, which has been a key partner with us. So tafenoquine is working well, single administration, single pill, but it can only use in G6PD normal patient. If you have a deficiency in G6PD, you cannot use tafenoquine. So in discovery, we're trying to develop the new generation of anti-hypnozoid drug, and we currently have 2 series in our portfolio, which are the 2 first ever non-8-aminoquinoline drugs to act on hypnozoites. We're forcing a clinical candidate declaration this year for at least one of the 2 series, meaning that in the next 2 to 3 years that we should start to get into clinical studies for those new drugs. But we continue, of course, the implementation of tafenoquine. I'm not going to go through all the line on these slides, but we want to continue, and there is a lot of work on pediatric tafenoquine. I won't talk about that because I know that there is a talk on Monday, if I remember correctly. We also have a study in Peru to try to look for mass drug administration. We also have a study in Suriname, and not Suriname, sorry for the typo. And we're also trying to explore and collaborate with other partners to try to bring tafenoquine outside of South America, because it's heavily used in South America, but we would like it to go and be used in other areas, in particular in Southeast Asia. Sorry. So this is it. It's a very speedy way of looking at the MMV portfolio. So I'll be here for 3 days. If you want to come and discuss any part of the portfolio, any drugs, you want to have information, please reach to me. I'll be around for all the time. But this is the commitment we gave to our board of directors when we put this strategy together. By 2030, this is the kind of thing we want to achieve. For most of them, we are on track, but the next few years are going to be interesting, in particular when, we hope that lumefantrine resistance will not appear as quickly as we think it could appear. And we hope that some of our strategy will at least contain the current resistance generation to make sure that those new drugs that are in our pipeline arrive at the time where the resistance could be an issue. And with that, I just of course, thank the donor, because without them we couldn't do anything. And I just wanted to thank you again for welcoming me in Brazil. It's been a pleasure, and yeah, it's a great meeting so far. Thank you.
So I, like all others, want to begin by thanking Danielle and Caroline for this invitation. It's been a wonderful meeting, and I'm excited to tell you about my lab's work to translate some basic biology discoveries into potentially pre-exposure prophylaxis against malaria. So this audience doesn't need introduction to pre-erythrocytic stages and how, although they are asymptomatic, their success at this stage is absolutely essential for the parasite to establish a niche in the human host. We know from all the various vaccine studies and various epidemiological data that inhibiting sporozoite invasion alone reduces both the severity and incidence of disease. So for this reason, we believe that combining drugs that target pre-erythrocytic stages with drugs that target erythrocytic stages could offer a valuable tool for controlling malaria. So towards this end, my lab has been interested in understanding the basic biology of pre-erythrocytic stages and, where possible, translating them into drug discovery programs. So the pre-erythrocytic stage begins when a sporozoite, shown here in red, invades a human hepatocyte. And as you can see, this is a very active process on the part of the parasite. The sporozoite is motile, enters the cell surrounded by a vacuole. In doing this, the sporozoite is responding to environmental cues that it is changing into physiological responses using a suite of second messenger signals. And my lab has studied the role of cyclic GMP, one of these second messenger signals in this pathway. So we focused on the major mediator of cyclic GMP in Plasmodium, and that's a kinase that we call PKG. This is a protein that we find to be expressed throughout the pre-erythrocytic stages in sporozoites and also in developing liver stages. By immuno-EM, we were even more excited to find that the protein is often present in micronemes, the organelles required for invasion of host cells. So it was guilt by association. It appeared to be in the right place at the right time. The parasite enzyme also has some interesting differences compared to the mammalian counterpart that could offer a pathway towards selective inhibition of the parasite enzyme. So the mammalian enzyme, for example, has 2 sites for binding cyclic GMP. The Plasmodium enzyme has 3. The mammalian enzyme acts as a dimer, whereas the Plasmodium protein stays as a monomer when active. We also had access to another really useful tool, this small molecule tri-substituted parole that I'll call TSP, that had been discovered by Merck research labs in a medicinal chemistry campaign against some chicken parasites. And the Merck group had also shown that this molecule inhibited PKG from Plasmodium falciparum very well. So we started by testing the effect of this compound on invasion by falciparum sporozoites, saw that it inhibited entry of sporozoites, falciparum sporozoites quite well. And it also had an effect on cynomolgi schizons and hypnozoites formed in primary simian hepatocytes. Another important difference between the parasite enzyme and the mammalian enzyme lies in the identity of the amino acid present at a key position in the kinase, and this is known as the gatekeeper position in the ATP binding pocket. In the Plasmodium enzyme, this is a threonine, which has a short side chain, and what that means is that the binding pocket in the parasite enzyme is quite large. In mammalian enzymes, including human ones, this site is occupied by bulkier amino acids with longer side chains, and so the binding pocket in the ATP binding pocket in the mammalian enzyme is much smaller. And what that means for compounds like the tri-substituted parole I showed you is that in the parasite enzyme, it lies deep into the ATP binding pocket, and this is modeled here in gray, is the molecule as it lies inside the parasite enzyme. It competes effectively with ATP for binding, and thus inhibits enzyme activity. Whereas the same molecule, modeled here now in orange, as it would sit in human PKG. Because the binding site is smaller, the molecule is pushed out of the binding pocket in the mammalian enzyme, and therefore is not as effective in inhibiting the human counterpart. So we made use, we exploited this difference by generating parasites in which the gatekeeper threonine had been replaced by a bulkier residue like a glutamine. Now, wild-type sporozoites glide in a circular fashion when you put them on a glass cover slip, and this motility is essential for them to invade host cells. When these wild-type parasites are treated with the tri-substituted parole, they can barely attach to the substrate, and they just drift along in media. So as a result of this decreased gliding motility, the wild-type sporozoites show a dose-dependent decrease in their ability to invade host cells, and consequently in the number of infected liver cells you get. In contrast, the mutant parasites, the ones that carry glutamine in the gatekeeper pocket, are much less sensitive to inhibition. They continue to glide in the presence of the compound. They continue to enter host cells in the presence of the compound, and infection by them is a lot more resistant in the presence of compound. So what these data showed us is that the ability of TSP to block sporozoite infection of hepatocytes was indeed being mediated by its inhibition of PKG. So PKG was the target in the parasite of this compound. And therefore, also that PKG is essential, plays a really important role in the parasite's ability to enter host cells. So we had established that PKG is required for the first step for entry, but we knew from our expression studies that the protein continues to be expressed throughout the liver stages. So we wondered whether it had additional functions at these later stages. And for this, we used the Plasmodium Berge model, where the pre-erythrocytic stages are much more experimentally accessible. So once the sporozoites have entered host cells, so here are some really nice videos from Volker Häusler's lab. The sporozoite turns into a trophozoite. It will undergo a series of nuclear divisions. Eventually, the cytoplasm will segment to form individual hepatic merozoites. These hepatic merozoites leave the infected hepatocyte in a vesicle known as merosome, surrounded by the host cell membrane, that are extruded directly from the infected hepatocyte. There you see one go into the bloodstream. And we can model the same phenomenon in tissue culture. So what we tested is different time windows of sensitivity to TSP post-infection by sporozoites. So we added the compound at different time points after sporozoite infection, and we saw that a time window corresponding to late liver stage development was sensitive to inhibition by TSP. So when the compound was added about 48 hours post-sporozoite infection, there was a dose-dependent decrease in the number of merosomes in the media. In contrast, the gatekeeper mutant, the one containing the glutamine, continued to produce merosomes in the presence of compound. Now, we confirmed these findings using a genetic approach. We had generated conditional knockout parasites in which the protein is lost in liver stages. And these conditional knockout parasites, sporozoites are able to invade hepatocytes. They form liver stages, but we never see merosomes in the media. So the parasite stays trapped within the hepatocyte, much like the pharmacological inhibition of the kinase. So what these data allowed us to do is to conclude that PKG had a dual function in the parasite, in the pre-erythrocytic stages. It is required both for the parasite to enter and for it to exit the hepatocyte. And through this, it provided biological validation for Plasmodium PKG as being a potential drug target for inhibiting pre-erythrocytic stages. And the fact that we could have the same effect using a small molecule inhibitor also allowed us to validate this as a pharmacological target. So the reason we thought of this is because of, at the time, the TCP4 indication by MMV, where they were looking for molecules with activity against hepatic schizons with mechanisms of action distinct from drugs used for treatment. And such compounds as, repeating very quickly, would be used for vulnerable populations such as pregnant women, potentially for local outbreaks where populations have lost or decreased natural immunity due to lower exposure, and the usual suspects of tourists and business travelers, etc., from non-endemic regions into endemic regions. So why do we think PKG inhibitors could potentially fulfill this requirement? So we believe liver stages are particularly vulnerable to Plasmodium PKG inhibitors because of the dual function, temporally distinct dual function of PKG in these stages. The same compound could inhibit sporozoites from entering hepatocytes. Even if some got through, the same compound would have a second opportunity to act by preventing the liver stages that do develop, by preventing them from exiting the hepatocyte. Further, in a single round of infection, the liver is infected only once, unlike the blood where there are, of course, numerous cycles of infection. And also the burden, the number of parasites transmitted by mosquitoes is thought to be less than 100. Contrast this with millions of asynchronous egress and invasion events that occur in the blood. So we thought PKG inhibitors might have value, with the appropriate characteristics, of course, might have value as inhibitors of the pre-erythrocytic stages. So with this in mind, I started a collaboration with medicinal chemists at Rutgers, Jacques Roberge and John Gillerin, and our aim has been to develop highly potent, selective, and safe inhibitors of PKG, while daily being reminded of what Bill Campbell said, that path from an active substance to usual medication is difficult. What Jacques' group has done has been to interrogate the TSP scaffold at various positions. And here to summarize several years of work, they've hit upon a series of molecules that show some important improvements. Compared to the parent molecule TSP, they have better cellular activity, and this is now against falciparum blood stages and also against sporozoites. And this improvement in cellular activity is not coming at the cost of reduced specificity for PKG. These compounds continue to be highly specific for PKG, more specific than the parent compound by comparing with the gatekeeper mutant. We tested these compounds against some current isolates that are resistant to currently used antimalarials, and they continue to show activity against these various strains, which provides some assurance that there's the low likelihood of cross-resistance to current drugs. So that's good. We then tested one of these molecules against falciparum sporozoites and also cynomolgi as a model. This compound has quite decent activity, worthy of continued development. So we were pleased about this. These compounds also have quite decent in vitro PK data. And we are particularly pleased about the in vivo ADME properties. This compound has a long half-life, almost 12 hours by IV injection in mice, and close to 9 hours orally in mice. And as a starting point for a molecule that we hope to be, that one hopes can be given once in 3 months or once in 6 months, we thought this was a good starting point. It's also highly bioavailable. So we next tested the efficacy of this molecule in mice. Since we have a pre-exposure model, the mice were pre-treated with the compound before infection by sporozoites, and then we monitor for development of parasites in the liver and in the blood subsequent to that. And what you see here is that all the vehicle-treated mice show clear signs of parasites in the liver within 48 hours, but the compound-treated mice, even 5 days post-infection, have no sign of infection. And in fact, we monitored these mice for 3 weeks in the blood, and we detected no parasites at all. The parent compound TSP also shows some activity, although this was not statistically significant in this experiment. So this was, in this particular experiment, we had actually given 3 doses of the compound, 2 prior to exposure to prevent sporozoite infection, and then 1 36 hours after infection, with the idea that if parasites do escape the first block, we could block them in the second round from leaving the liver. So after this experiment, we wanted to test the efficacy of the molecule, of a single dose of the molecule. And that's what we did by giving 1 dose of the molecule prior to sporozoite infection, either intravenously or orally. And we were happy to see that all treated mice once again stayed negative for a month after sporozoite infection. So this compound was orally efficacious at a single dose in preventing infection of mice by Plasmodium burgii. So with this, we felt that we had a good lead molecule. And so now what lies ahead is optimization of this lead. And we are trying to take sort of a rational approach to this. We have solved the structure of a couple of the compounds bound to Plasmodium vivax PKG. Vivax PKG, fortunately, is nearly identical to falciparum PKG. So we expect the molecules to have similar activity against vivax. And in collaboration with Schrödinger, we've also developed a computational model of the compounds as they are binding to falciparum PKG. And what I show here is just a superposition of the computational model of falciparum PKG with the X-ray solved structure of vivax PKG, and there's very good agreement between the experimental structure and the computational structure. So we can use these data now for prospective design, rational design of inhibitors with greater potency against the target. So another challenge that we face, but one that we think is tractable, is the selectivity of the compound for the human kinome. The compound is extremely selective against human PKG. It does not inhibit human PKG, but it does hit a couple of other human kinases of concern. And the 3 major ones happen to be something called CIT, ROC1, and ROC2. They're all related. That could be a good thing because if we de-risk one, we think we will de-risk the others. And our preliminary data suggests that that is indeed possible. So RUPB69 is a newer molecule in this series. And we were really happy to see that not only is it almost 10-fold more potent against the target, against falciparum PKG, it is also far less potent against the human off-target. So we can improve potency while improving selectivity. And I think if we can do that, I hope I can convince you that if we are able to do that, then small molecules inhibitors of Plasmodium PKG could be used to block pre-erythrocytic stages and provide chemoprotection. And that's where our ongoing work is focusing on improving potency and selectivity properties while maintaining all the favorable PK that we have so far. With that, I'd like to thank a lot of people who've helped with the work. A lot of current and past members have contributed to this. A lot of the work I showed you was a PhD student, Hannah, research technician, Anusha, and a postdoc fellow, Aminata. I already mentioned this is a close collaboration, medicinal chemistry with medicinal chemists, Jacques Roberge and John Gilleran. We collaborate with Alison Roth at Walter Reed for testing against falciparum and cynomolgi sporozoites. We collaborate with SSGCID for the structural biology and with Schrödinger for the computational biology. And finally, of course, nothing is possible without funding. And thank you all for your attention.
É dela. Yes, yes it is. Acho que embaixo. Oh, okay. Can you hear me? Because up there we cannot hear so well. So thank you very much for staying up to this last session. And I'm pretty sure that you are all tired, and I'll try to be as clear as possible and not so boring. So thank you. First of all, I would like very much to thank Caroline and Daniel for inviting me to be here in this special day for malaria, and it is very special to me as well. So thank you very much, Caroline and Daniel. So my name is Rafael Guido. I'm from USP at the campus in São Carlos, which is the countryside of this state in São Paulo. And there we have, I'm from the Physics Institute of São Carlos, and there we have a lab where we can cultivate the parasites within the Physics Institute, which is not so friendly. They do not like so much, but they had to bear with me. So, and then we cultivate the parasite, and using this cell, we can build a whole cell assay to screen for compounds from both natural or synthetic compounds from natural or synthetic sources as inhibitor candidates. And as we find some inhibitors, we can look at the structure of those inhibitors, the structure of the molecule, and then we can develop and try to design new and improve inhibitors using the information about the molecular structure of the ligand. Therefore, we can use ligand-based drug discovery strategies. And when we do that, we collaborate with our very good friends from the organic synthesis, and then we can build what we call the structure-activity relationship, where we can modify the molecule and then test the molecule again against the parasite and see if we can improve properties such as potency, solubility, and other DMPK properties, for example. At the same time, we can use the cell culture that we have in the lab and then look for specific genes that we want to investigate as molecular targets for drug discovery. And then as long as we can clone and purify the product of those genes, we can have a pure protein, which is usually an enzyme, and then we can build an enzymatic assay to screen for inhibitor as well. And when we do that, we can, of course, find the inhibitor. And by doing this, now we know the target of the inhibitor, or at least one of the targets of the inhibitor that we are investigating. And in parallel, we can conduct structural studies to investigate the molecule from a structural perspective to understand what is the 3D structure of that specific biomolecule. And by doing that, we can use X-ray crystallography or cryo-EM now. And when we can solve the structure, we now know the binding site of that specific molecule, and we can use that information to do structure-based drug discovery, as Purnima just showed us. And this is very interesting because now we can look at the target and design new inhibitors in order to improve the activity that we want by modifying the molecule and test it again against the enzyme or against the parasite at the same time until we have a compound that is what we call a candidate for in vivo studies. And then if the molecule succeeds in these in vivo studies, we have what we call a lead candidate that we can start to investigate in more detail in order to develop that molecule as a possible drug candidate in the future. So over the past 10 years, our group in São Carlos has been contributing, discovering several new chemical series as lead candidates for malaria. And the ones that I selected to discuss with you today are two recent examples that we had published recently, and both of them, they came from nature. We were inspired by nature because we in Brazil, we have a very big biodiversity, and it's very interesting to investigate our biodiversity here. So the first one, it was a molecule that was extracted from a marine sponge collected here in the shore of Brazil by Professor Roberto Berlink from the Chemistry Institute in São Carlos, and he purified them, isolated and purified these molecules and sent to our lab in order to us to understand if that molecule could be a potential inhibitor of the parasite. So we test that. Giovanna Mendes, who is my PhD student, she screened those molecules, tested those molecules, and we could verify that the molecules were indeed Plasmodium falciparum inhibitors, the sensitivity strains in the 3D7 with submicromolar activity, which is quite good for a molecule that do not pass for any improvement yet. So the molecule also showed moderate cytotoxic activity against the HepG2 cells, which showed to us that they had promising selectivity index. Next, we test the compounds against the representative panel of resistant parasites that we have in the lab, and we learned that the molecule did not show any cross-resistance with the standard antimalarials. Next, we wanted to understand more how those molecules were killing the parasite, and then we performed a speed of action study when we test the molecule over from 24 until 72 hours, and we used two controls, one a fast-acting inhibitor, which was artesunate, and a slow-acting inhibitor, which was pyrimethamine. Those molecules, as we can see here, the natural products, the benzaldehydes, they show a profile that it was closer to the fast-acting inhibitors, indicating to us that those molecules probably were fast-acting inhibitors of the parasite. To confirm that, we perform a morphological analysis, and here we use the same time frame, and then we have in here, for example, at the 0 hour, we have the parasite in the ring form, and then we follow after that the trophozoite, and then 48 hours later, we have a new red blood cell infected by the parasite, by a new parasite. When we see that with our positive control, the artesunate, we can see that at 0 hour we have the ring stage, but 24 hours later we have this parasite that could not develop. So this is characterized by those pycnotic nuclei. The parasite here is suffering, and as we can see, the parasite cannot develop anymore in the presence of this specific positive inhibitor. And when we look at our test compound, which were benzaldehyde L and F, we could see that at 0 hour we have the ring stage, and 24 hours later we have those pycnotic nuclei, as we could see for the positive control for confirming to us that those molecules were indeed fast-acting inhibitors of the parasite. So next, we want to understand if those molecules could also inhibit not only the parasite that we have within the lab, but the circulating strains that we have here in Brazil. So there, we sent the compounds to Porto Velho. Caroline did those results, those assays, and we could realize that yes, the molecule had potency against both Plasmodium vivax and Plasmodium falciparum isolates. It was not so potent as was against the lab strains, but it was good enough to show us that molecule was active. And then we want to verify if the molecule could inhibit the parasite within the animal. So we use the PfPKG model using to evaluate the efficacy of the compound. And we treated the compound, the animals for 2 days in here, because we did not have so much molecules because this specific molecule was extracted from nature, and the molecule at day 5, it showed 94% of reduction in parasitemia, which it was quite good, because this molecule, as you may remember, did not enter any specific development yet. So it indicates to us that this molecule is indeed a molecule that we could continue to investigate as a possible lead candidate for malaria. And on top of that, the treated group, we could see that the animals survived until the end of the experiment, suggesting to us that the molecule was tolerable by the animals and protect the animals from the infection. And due to the relevance of those results, this work was selected as the front cover of the ACS Infectious Disease, the last July issue in 2025. So I recommend, if you want to know more about, please access the link here. The paper is very good. In fact, I know that it's my child and it's very, very beautiful, but I really recommend you to read it. And the next steps for this series is that we, knowing the structure now, we want to develop these molecules probably as a lead candidate for malaria. And in order to do that, we made a collaboration with Professor Igor Djurberg from the Chemistry Institute in Campinas. And he had already synthesized over 200 molecules. And we simplified the molecule because, as you could see, the molecule was quite big. And we realized that we have 5 series now working in parallel, and we could realize that as we simplify the molecules start to behave differently against the parasite, and we now investigate that in order to understand the mode of action of those molecules. And the second example that I would like to share very briefly with you was conducted in collaboration with Professor Carlos Roque and Professor Carolina Aguiar, which were another molecule extracted from the sea, but this time which was extracted from a gram-negative marine bacteria. And this marine bacteria produce as a natural product these marino-quinoline derivatives. And Professor Carlos Roque and his team, they synthesized the molecules within the lab, the natural products, and then we could test those compounds and confirm that those molecules were indeed inhibitors of the parasite with very good ligand efficiency because the molecule was small but very potent from a hit perspective. So we started to investigate doing new synthesis, modifying the molecule as you can see here, and then we could establish the structure-activity relationship for this series. For example, when we substitute the put a methox substituent in here, we could improve the potency of the compounds. And next, we use molecular modeling to get new ideas and new insights how to modify the molecule, and then we could deliver or identify improved inhibitor with low nanomolar activity, which were tested again against the stage-specific assay, using stage-specific assay to try to understand what is happening here. And we realized that the molecule discontinued the development of the parasite after 80 hours, suggesting that it could be a fast-acting inhibitor. And indeed, it was. We did a collaboration with GSK. They tested the compound and confirmed that it was a fast-acting inhibitor. And also it has a prophylactic potential because it inhibited the liver stages of the parasite in a low nanomolar range activity. And we tested against a resistant strain, which was quite good as well, and also against the field isolates, suggesting to us that, yes, this molecule is something that we can still working on. And so far, what we learned was that the molecule had dual stage activity, which could be very interesting, as we just showed before. And of course, we wanted to understand if the molecule has in vivo activity. Yes, and the answer is yes, the molecule has in vivo activity. It showed a reduction of 70% in parasitemia at day 5, and it was sustainable until day 9 and until day 9 in this experiment. So far we learned that the molecule, yes, it was good, but we wanted to know more because we wanted to know the mode of action of that specific compound. And in order to do that, again, we synthesized new molecules, and those molecules had the fluorescence property that we used to investigate the possible mode of action of the compound. But before we verified that the new derivative, it was potent as well. It has improved, as we will see later, the pharmacokinetic properties of this compound because we were aware of that we need improvement. The molecule was active against a representative panel of resistant strains of the parasite. And also the molecule showed in vivo activity in the PfPKG model, showing a reduction of 96%, which was better than before, the previous compound. That is mostly because we could improve the pharmacokinetic properties of the molecules, such as solubility, the plasma stability, the metabolic stability, which increase the half-life of the compound, indicating to us that, yes, we still have room for more improvement and that we are probably in the right direction. And as we want to know the mode of action of this theory, we took advantage of the fluorescence properties of these molecules, and we saw that it showed increased fluorescence intensity within the digestive vacuole of the parasite. As you can see here, it was significantly different than the cytosol, and by doing that, we were probably suspicious that maybe it was inhibiting the proteolytic activity of the digestive vacuole, and we tested that, and we could see that at 10 micromolar, all the proteolytic activity was abolished. And then against cysteine protease, which is the main protease that we have, one of the main proteases that we have within the digest vacuole, we could see that a decrease in inhibitory activity of the enzyme. And by knowing that, we could see that, for example, this is this one. This is the main protease that we have within the digestive vacuole, which is the falciparum 2. And as we can see, this molecule has a bind site, which could fit a molecule. And by fitting this molecule here, it is blocking the substrate from binding to it. And by knowing that, we can see that there is complementary between the molecule, the small molecule, and the biomolecule. And there is still room for further development in order to look at the bind site now and try to design new inhibitors with improved properties. So, it's not this one. Where is this one? And just to finish my presentation, I would like very much to tell you a very important and fantastic project that we have in the lab. That is the Center of Excellence for Brazilian Field Isolates, which is a project that I have the honor to lead and share the leadership with Caroline here and Delio, that is back there. And we work under the guidance of Brice. So Brice is my boss here. So yeah, I'll try to do my best. Brice. And here we, as a center of excellence for MMV, we evaluate new candidates for malaria, most preclinical candidates for malaria from several different pharmaceutical companies that we have. They are interested to develop new molecules for malaria, which is very good. And by doing these evaluations, we have so far tested over 150 molecules against the field isolates, both from P-falciparum and P-vivax, and which is very nice because we are, as Brazilian, contributing to drug discovery for malaria, collaborate with people all over the world under the guidance of MMV, which is a fantastic entity for the development of new medicine, as we could see. And by doing that, we also contribute to science and doing very good results, collecting very good results and publish those results. That is for a disease, a tropical disease that impacts most of poor people in the world, but we can do state-of-the-art science and publish those results in very good journals. So I'm very proud to work with my team and Caroline, and it's very nice to be here. So I would like to thank you all for your attention, to stay until now. And of course, the last but not least, I would like very much to thank my team. Those are the very important person because they work a lot at the lab that allows me to come to Campos do Jordão and enjoy the city and present their results. So that's why they are very important. So it's very important to thank them. So, and of course, I will be very happy to take questions if there's any afterwards. Thank you very much for your attention. On time. Obrigado.
So last talk of the day. I'll start also by thanking and congratulating Daniel and Carol for organizing this beautiful meeting. It's such a pleasure to be here, and it's also an honor to divide this session with Purnima, an old friend from 20 years ago in Victor Nussensweig's lab, and also Rafael, that started his career in malaria because I started because of me. So I'm really happy. So without delay, let's start. Can I have my presentation, please? I don't have my... Oh, it's there. Okay, I'm the one that needs... Okay, so in our lab, we have been working, I don't need to show for this audience the malaria cycle, but just I would like to highlight to you that our lab, among several other labs, have been publishing work showing that during the cycle of malaria parasite, it has a close perception of the microenvironment. So the idea of working with this relation with the microenvironment with Plasmodium is to understand, is to decode the signaling pathway that coordinates all the process that takes place during this very complex cycle of malaria parasites. So the lab has basically two focuses. One is to identify molecular and cellular pathway at the cell cycle, but also do some drug screening. And I just highlight some of them to you. I won't have time to show these are published data. You can follow some of them. Also, one of them with Carol and Rafael that was mentioned here just in the previous talk. Okay, so the question we ask ourselves in the beginning is, what sparked second message in the signaling generation parasite? So you hear before Purnima Bhanot showing the PKG, the relevance of PKG, so such a downstream signaling pathway. We want to understand how these signaling are starting in the cell, in the host parasite. So which pathways drive them, and do they shape, do they have any responsibility of our parasite survival? So having said that, we have been working for many years with calcium signaling. Why calcium? Because calcium is one of the most important ions for coordinating signaling pathways in all eukaryotic cells. So we try to understand if that is also happening in a parasite that lives inside another cell, such is the case for Plasmodium. So a very talented PhD in our lab, Lucas Borges, has generated the genetic calcium sensor, a G-Camp, that is based, GFP is linked to a piece of calmodulin. And once this calmodulin binds calcium, cytosolic calcium, it increases the fluorescence in the cell, so you can follow the fluorescence and understand if calcium is involved and it's increased in the cell. So we have been using these transgenic cells to understand signaling pathway, but also to screen drug compounds. I'm going to show you some of the data. So here, this very complex signaling is just, I can summarize in one sentence. It's called a very spontaneous calcium oscillation in the cells. So usually cells do that to tell the cell which genes should be transcribed, should be activated in that moment. So we find out using this tool, the GCamp, that Plasmodium also present a spontaneous calcium oscillation. But this was not easy to prove. We have to collaborate with a mathematical, Rogério Hashimoto from USP, to apply a Fourier transform and show that this is really an oscillatory pathway. However, more recently, another very talented girl, PhD student that is doing part of her PhD at Rutgers, at same university as Purnima. She is using this GCamp to show oscillatory behavior in Plasmodium when the cell is removed, is out of the red blood cell. So this is just a parasite. And she was able to show very nicely a huge oscillation of the parasite when calcium is in the extracellular medium. So she's also interested in studying the process of merozoite invasion in a grass. So she made this very nice system to stick the parasite under an agarose gel to observe the process of egress of the parasite at the end of the asexual stage. So here you can see clearly that when the parasite, when you remove calcium from extracellular and by adding a GTA, this oscillatory behavior stopped. So here we have a very quick movie. Is that, can you start it? Or maybe, okay. So you can see the merozoite. So this, again, this is the GCamp, the fluorescent parasite that express, it's showing. So what you see in the rise in the fluorescence is a rise in cytosolic calcium. So the parasite has a burst of calcium increase just up the moment before egressing the cells. So as I mentioned to you, this is a very interesting process to understand, to understand all this behavior. So, but then the question that we ask, okay, this calcium is generated, the parasite is able to keep it at a very low homeostasis, but how this is activated in these cells? So usually when you think of any eukaryotic cell, you think of the membrane, the PLC membrane that generates, activates an IP3, and the endoplasmic reticulum has a receptor that will couple to this calcium release. Okay, so but in Plasmodium, as you know, in the genome database, there's no receptor for IP3. So this is just a recent work that, just make sure that I don't follow. There will be a disaster. Okay. Okay, so this was the first demonstration that, in fact, if you use a caged compound in flash photolysis, you promote the IP3 generation, and that is able to increase the cytosolic calcium in an intact parasite, inside the parasite. Also, this is an old data from our lab showing that the parasite, upon addition of an agonist and melatonin, is able to promote the calcium increase that's blocking the presence of a PLC inhibitor. We showed many years ago that the parasite resides inside the parasitophorus vacuole. This membrane, this space of the membrane is responsible for create a very high calcium atmosphere microenvironment. So more recently, Eduardo Alves, a PhD student in our lab, he did a very nice experiment by doing a chromatography IP3 for IP3 binding and passing through the lysate of trophozoite stage parasite, and we come up with one candidate here for the IP3 receptor, which is the MGR. Yes, this is the MGR that you know, the multi-drug-resistant gene that we are proposing that is the IP3 receptor in the parasite. I won't have time to show you detail, but this is published at the current Research in Microbial Science. And just to highlight that one of the partners of the MGR is REC1, and REC1 is a well-known coordinator for IP3 receptor in mammalian cell. So it's just a very exciting finding. Okay, so then it comes to the question, what about the upstream signaling pathway? So I'll show you some of the possibility for raising cytosolic calcium or IP3, but usually cells have GPCR, G protein coupled receptor, at its membrane that is responsible for sensing any sort of peptides or ions or endorphins or pheromone or thousands of myriads of compounds. So, but why do we care about GPCR? Why is it important to uncover GPCR for Plasmodium? If you look at the market, 40% of all the pharmaceutical drugs in the market comes from targeting GPCR. So you find many drugs in the market that target GPCR. So we found that that could be very interesting. And by searching in the genome, we didn't find by BLAST any candidate for GPCR in the Plasmodium database, but by searching for protein that has 7 transmembrane domain, which is the signature for GPCR, we found 4 candidates, SR1, SR10, SR12, and then SR25. I'm just going to show you some of the data we have from SR25. So Luciana was the PhD student involved in the identification of this GPCR that is expressed during all the sexual stage. But before telling you about the SR25, I just want to highlight a very interesting paper that was published by Chetan Chitnis from Pasteur Institute. He showed the merozoite, as you know, the parasite experience very different microenvironment. So if you think of ionic composition, it's very difficult on the point of view of potassium because you have a very high potassium concentration inside the cell and very low potassium concentration outside the cell. And it's the opposite for calcium. Calcium is much more higher at the outside. So what Chetan did was subject the merozoite to a shift of potassium concentration, because when the parasite is aggressing, it's subjected to this shift of potassium concentration. So he measured a rise in cytosolic calcium when the parasite was subjected to this condition, and then he's proposed that the parasite, this is used for the parasite in the moment of invasion for regression of some of several contents in the apicoplast. Okay, so having said that, we try we construct a knockout of the SR25, and we subject the parasite to a shift of potassium concentration from high to low in a different conditions, and show that the Y type is in fact able to increase the cytosolic calcium in the presence of this shift of potassium, but not the knockout parasite, the SR25, as you can see here at the left. So, I also don't need to explain to you the relevance of hemozoin, the crystal of hemozoin for these parasites, but also what we found interesting is a phenotype of this knockout parasite. If we treat with piperacin for 2 hours the knockout parasite, we observe a reduction of the crystal of hemozoin compared to wild type. So what we learn from this knockout parasite is that piperacin and lumefantrine, the parasite is much more susceptible when it's missing this SR25 to lumefantrine and piperacin. As you can see, the IC50 for both antimalarial drugs. So we come up with a very interesting idea and model that the parasite has in fact a sensor at this membrane that is able to sense this shift in potassium concentration, increase the calcium concentration, and then for somehow this is linked to the digestive vacuole. And just to highlight to you that in parasite, this is the most important site for calcium to the parasite to keep calcium. Okay, so we did also for triazole, a series of triazole compounds. We found some susceptibility for the knockout, which is a small susceptibility. And more recently, we Wahid, a PhD in our lab, he did also, in collaboration with Mateus Pessenin, a screening for the TCOMS library, the 13,000 compounds, and we select some of the compounds that was unknown target and did a dose response at IC50, and we find a very interesting IC50 lower than 100 nanomolar, also very good gametocyte assays with a very low activity. And we also did some calcium signaling assay for this parasite. So this is going to be presented today. If you have some time, you can see in more details all of these data that I'm just showing to you, that I'm going to highlight here that what is interesting in this calcium data, again, using the GComp, the fluorescent parasite that I will show you that express this protein that is able to sense calcium, that if you remove calcium from the ER and then afterward you add the compound, you don't see any calcium, any rising in calcium. So that is telling you that this drug is able to signal and then remove calcium from the ER. So this is a very interesting data. I think I won't have time to show the other part, but it's just to highlight what we found. We think the PLC-IP3 calcium signaling is operative in Plasmodium falciparum. That data not only for our lab, from several labs. The GComp is an interesting tool, so you might consider to use. I'll be very happy to share with you. And also, the SR25 is the first GPCR identified in Plasmodium. And the knockout parasite is interesting as it's more susceptible to some antimalarial drugs. So you can see the 3 posters. Mariana is going to present tomorrow, but Abby and what he is going to present, is presenting today. Okay, I think I'm going to skip this very interesting data that we have also recently in our lab, the identification of MORC, which is the protein that's responsible for the switch of gametocyte, and it's mostly at the nucleus, and more recently, in collaboration with synthetic chemistry, we identify some of the HDAC inhibitors also that hits gametocyte stage and the sexual stage, and we show by the activity of the recombinant enzyme that was the target. So having said that, I'm keeping the time, and I'm very grateful to all the collaborators, the postdocs, and the PhD students that I had the privilege of working during this time. The first line are the current members in the lab, and also I was very lucky to collaborate with people from all over the world in different universities, and also here I would like to thank PfPKG because this is also a great funder for our work, but also CNPq, CAPES. This was a collaboration with the GSK in a grant awarded by NIH, together with Andrew Thomas, our collaborator at Rutgers. I think I made on time. Time is over. Thank you so much for your attention.
Hello. Yeah. Well, I will take advantage of my position here and ask the first question. I would like to ask a question to Brice. Now I'm there. It would be nice to introduce myself. I am the head of clinical research platform in Fiocruz, but I have been working with the NDI for more than 20 years, and it's great to see this new model of PDP working, and MMV is another example of this. I'm also very happy to see that all of you know each other, meaning that collaboration is the only way to do research nowadays. You have been giving us good news about, so it's two questions. One is you have been giving you good news about long-acting drugs and a single dose scheme, if you want. So how do you envision to see MDA, a mass drug administration of a single dose and a long-acting injection? It's because TB Alliance is working in something like this. It's just a kind of having people being treated, and then for a while they have been keeping on that. And the second question, it's about pregnant women. It's a really neglected population. And how early are you including reprotox in your screening? And if it's go but non-go results, because this would increase your attrition rate. But of course, it's super important. Otherwise, we are never going to have a drug for pregnant women.
Okay. Thanks for the question. I think the first question, because it's hard to hear very well, is about mass drug administration using single-dose cure treatment or LAI, right? Yeah. Well, yeah, I don't know if I can give you a scheme, but we're thinking about mass drug administration, but in order to do that, you need to have a very safe drug, of course, and that is safe in every population, in particular pregnant women and lactating women. So I think at the moment we don't have this drug, so we're far away from that. So the way we're going to approach that is more, rather than mass drug administration, is administration in specific population in specific cases. Of course, if you have a very safe drug with all the attributes that we were listing here in terms of LAI or single dose cure, then you can start thinking of mass drug administration. But I don't think we have the drug for that today. In terms of pregnant women, it's a good question. We have an initiative called MIMBA, so Mother in Malaria and Babies. No, Malaria in Mother and Babies, sorry. So the goal of this initiative within MMV is to try to bring this population at the forefront of drug development, so include them as soon as possible in clinical trials, but also bring teratogenicity assays as soon as possible. So, for example, at the late stage or clinical candidate stage, which is the stage at which we declare a molecule before it moves into GLP studies, we are now requesting those assays, whether it's in vitro assays or in vivo assays, to be run for teratogenicity in order to understand the potential of the molecule for pregnant women and women of childbearing potential. So we're bringing that as early as possible. One of the main issues that those assays are not highly predictable, so in particular the in vitro assays. So we're working on that internally with our tox experts to try to build some kind of correlation. So we're using, for example, the zebrafish assays or human stem cell assay. So we're building a correlation, trying to understand if those assays are predictive of what's happening, at least in animal studies, because that's the one you want to predict, because they are the filter after that in order to give you a label or not on your product. So yeah, we have that in mind, and we're bringing those populations as early as possible. One last thing you said is it a killer or not if you are teratogenic positive? There's been huge discussion internally, I can tell you, because a lot of molecules are teratogenic. So you could kill half of your pipeline. We don't. We keep those molecules because they can still be useful in certain indication or population. But of course, if we have to prioritize, we will prioritize molecules that are safe in teratogenic assay.
Thank you very much. I don't know if there are any questions. Go ahead, please.
Very fast. Thank you all for your talks. I have a question to Purnima. So you showed some anti-hypnozoital side effects. So I was wondering why not taking the opportunity to meet the TCP3? First question. Then I saw that you acknowledged Alison Roth, so I assume that she ran those assays at Walter Reed. So the question is what drugging scheme did she use, like a radical Q scheme or a prophylactic approach to the liver stage assay?
I'll answer the second question first. So you asked whether in cynomolgi, whether it was radical cure or it was not. So this is prophylactic mode. Given
so 10.0, right?
Excuse me?
After sporozoite infection, right? After.
Prior to sporozoite infection,
okay.
So it prevents the formation of hypnozoites, presumably because it blocks invasion by sporozoites. Given what we know about the mechanism by which the target acts, there's no reason to believe that, we don't believe that it would kill already formed hypnozoites, although it might prevent the egress of hepatic merozoites from reactivated hypnozoites. We have not tested that though.
So my first question doesn't make sense.
Was the first question?
No, in that case, my question doesn't make sense because I didn't understand that you drugged before sporozoite invasion. So no way to meet TCP3 in this case, right?
So in the mouse efficacy studies, for example, the doses are given prior to sporozoite infection because what we are pursuing is pre-exposure prophylaxis.
Okay, thank you.
I would like to make a question. Oh, go ahead, please. Audience first.
I have a very simple question to Brice. You mentioned when you're talking about... Can you hear me now? When you're talking about tafenoquine, you mentioned about a next generation drug that would be safe to use for G6PD deficient patients. So is this just because this novel molecule will not be an H-aminoquinoline, or is there like an in vitro assay that you can use to measure?
Yes.
If there is such an assay, why wasn't it like used when developing tafenoquine, or was it? And like,
yeah, it's a good question. So the next generation, okay, they are non-8 aminoquinine, but we also know they are not, they don't have any hemolytic effect in G6PD deficient blood, because we have an in vivo assay. So it's a mice model where we engraft the mice. So it's a humanized mice model where we engraft the mice with G6PD deficient blood, and then we test the compound in the mice, and we see the number of red blood cells, if there is hemolysis or not. And we use tafenoquine and primaquine as a control. So we have this model, and every single clinical candidate that we push for, whether it's for radical cure or anything else, we test it in this model, because we want to make sure they're not hemolytic. Regarding the second part of your question, I guess at the time when they developed tafenoquine, I mean, it took 30 years to develop, 30 or 35 years to develop tafenoquine. This model was not available at the time. So they find it the hard way. So now it's, I would say it's easier because we have the model. And indeed, those two series that we have now, we tested in this model, and they're not hemolytic. So we hope that at one point they could replace tafenoquine, but it's, I mean, remember, it's in 10 years or 12 years time. It's not now.
Can I just do a second question? You mentioned about like a tovacone prodrug, which is highly synergistic with a different novel molecule. I did a quick search, and it doesn't look at all to Progonil, but it's still highly synergistic. Do you know if it's doing the same thing that Progonil does to be highly synergistic?
So it's a different, it's a BC1 inhibitor also, but acting on the QI side. So there's two sides on BC1, QO, QI. So those two molecules, I mean, atovaquone binds to the QO side, and ELQ, which is the second molecule, or 055, binds to the QI side. So they act on the same target, basically. So that's where we think the synergy is coming from.
Thank you so much.
A little over a month ago, the DD4GH platform, AI, was launched, which is drug discovery for global health. I don't know, Brice, if you can tell us more about it, and if there's anything about not only drug discovery, but also drug resistance.
Yeah, so it's, I'm not the best person to talk about it. I'm a biologist, not a chemist, so I apologize for that. So it's a new tool that we've been developing using Gates funding, where it's an AI model that everyone will be able to use. It's free for everyone. You log on the website, you enter your molecule, and then it starts to do some predictions in terms of activity, but few other parameters. I don't think we built the resistance in there yet. This is the long-term goal, but it will take some time. But the preliminary model that we have, you can already, I think it's already online, right? Yeah, you confirm that. And you can use it for your molecule and help you in making the next one and understanding better your SAR. So it's really using an AI model. That's all I can say about it. Yeah, Purnima, do you want to say?
We've tried this model a little bit in the last few weeks. It's very new. But what it helps you do is you feed in information about your molecules. It's better to start if you have data with about 50 molecules, so it can build a reasonable model, and then using data from background from previous studies that MMB has done and is fed into building the molecules, building their various models, is useful for predicting activity against cellular activity against 3D7. It's helpful in predicting cytotoxicity against mammalian cells, for in vitro PK properties as well. Those are the three parameters that we have tried so far. We are also actually trying it against, for example, like human off targets. We're just starting to do that. So yeah, I think it's a really useful tool, and I would give a shout out and encourage people to use it. It's also pretty user friendly. I'm not at all a machine learning expert by any means. I'm not even a medicinal chemist, but the team behind it has been very supportive. They're very willing to have meetings with you, talk you through the platform, and the learning curve is not very steep and not very time consuming. Thanks to MMV.
Hi, everyone. A little bit off topic, but pertinent, I think. So it seems like you all test the impact of these compounds on PF3D7, a strain that has been lab adapted for a long time. I heard that you also use some of these other, again, lab-adapted strains, UD2 and so on and so forth. And of course, you have to start somewhere, and you can't possibly adapt all possible strains to the lab. But I guess this is from a curiosity standpoint. Where does genetic variation in Plasmodium falciparum play in your pipelines? And it seems that at least once you know the mechanism of action, you can certainly leverage a lot of the known information of genetic variation to try to see likelihood of resistance emerging. So just curious how this is factored in.
So we've tried to do this since we have a very much a target-focused approach. We have looked at over 23,000 falciparum genomes for variation in the PfPKG gene, specifically around the gatekeeper site, because we were interested in knowing if there are already parasites out there with these kinds of substitutions that we could predict would be resistant. The good news is, at least of the ones that are in the database, the 23,000 or plus, none of them have this variation. The variation that does exist based on our modeling, that enzyme, if anything, should be slightly more sensitive to the because the gatekeeper residue has become even smaller. It's gone from a threonine to a serine. I don't know if that answers your question a little bit. We have not tested against field isolates of falciparum.
Yeah, thank you. No, it does answer my questions, but one of the issues to consider is, again, reference-based biases in terms of inferring the genotype. So all the genotypes that are available out there were obtained by mapping against 3D7.
Sorry, time is over, but I think that I would give Rafael the last words.
Thank you for your question. I think it's very important. That's one of the reasons that we test our molecules against circulating strains of the parasite, because we don't know the genetic background, but we know that they are might be different, but might be different from the ones that we have in the lab. So it's not directly addressing your question, but you use, I did not show the results in here today, but we use in vitro evaluation, evolution in order to generate resistant strains within the lab to try to anticipate what would be the mechanism of resistance for those specific parasites. So it's not so big genetic difference, but we can see that what are the problems that the molecule will cause on the parasite. So yes, selecting the molecules and the genetic variant as well. So thank you.
Okay. Yeah, just to complement on that, yeah, at MMV, when we select a candidate, we don't only use 3D7 and all the lab strain. All the candidates must show activity on field libraries across the world. So we have Center of Excellence, as Rafael said, here in Brazil for chloroquine and falciparum strain in Brazil, but we also have 2 of them in Africa, East and West Africa. So we do test and we make sure that the activity we see correlates with what we see in the field isolates, because otherwise it could be a worry. For example, we had compound where we saw a difference between Vivax and falciparum, and actually one of them we push up to the clinic, and indeed this was also confirmed in the clinic. It was really falciparum active, not active on Vivax. So we do that also.
Can I ask a question to the question? Or to, yeah, so what I wonder about these data is by the time a compound actually gets to market, the isolates have completely changed, right? So how much of a go, no-go criteria should this be? You're testing against strains in the field now. The timeline of development is much longer.
It's true, but remember when we develop the drug, then you move into clinical phases where you test drug into patients, which are in the area where you're going to use the drug afterwards. So your phase 3, which you're going to test several hundreds patients, it will be an area where later on the drug will be used probably a few years after that. So it's not going to be 20 years after. It's only like maybe 3 to 5 years after that.
Well, thank you much to our lovely panelists and the excited results. Sadly, we have to close the table. It's a very exciting topic, and we would like to invite you all to the reception dinner. Thank you for being here.
Para quem ficou até os bravos ficaram até o final.
For the courageous one that stayed up to the end, we are going to have dinner at Serra da Estrela Hotel, that is very close to